Hello everyone, and welcome to today's live broadcast, Basic Concepts in Transient Expression using 293 and CHO cells, presented by Henry Chiou, the Associate Director in the Life Sciences Solutions Group at Thermo Fisher Scientific. We are excited to bring you this educational web seminar presented by LabRoots. This event is sponsored by Thermo Fisher Scientific, and this live broadcast is brought to you by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Please join me in welcoming Dr. Chiou. I will now turn the presentation over to him.
Okay. Hello, and thank you very much, all of you who have joined me today for my presentation on basic concepts in transient expression from 293 and CHO cells. My goal for this presentation is just to give you, again, sort of a little bit of a primer, some basic ideas and understanding of transient expression systems, and sort of some of the ideas that will really help you in terms of thinking about how to best optimize and get the best performance out of your transient expression systems. So let me start off by just a really quick introduction here. You know, the expression, the production of recombinant proteins from mammalian cells has become an increasingly important tool for use in life sciences for obviously a variety of reasons as we delve deeper into trying to understand the structure and function of proteins and how they work in our everyday lives, as well as quite a lot of interest because more and more of the drugs and the therapies that are being developed now are based on the use of recombinant proteins. So, for example, antibodies that are targeted to fight cancer cells, or various types of molecules that are used to either stimulate or block different biological functions within our body are being based on recombinant proteins. So expression of these proteins, being able to get a good amount of them and being able to do research and work with them in the lab is a very important tool. And the classic method for being able to produce proteins in mammalian cells has been by the use of what we call stable expressing cells, where you transfect your gene that's expressing your protein of interest into the cells and go through a process to select for that gene stably integrating into the cell. And then once you have those stable cells, to expand them out to grow up as many as you need, then to produce the amount of recombinant protein that's necessary. However, that process it requires a fair amount of work and time to do, and it can take many weeks to months in order to create those stables. Obviously, once you have the stables, then ideally you can be able to produce as much protein as you want and repeatedly over many different expression runs. However, over the past about 15, maybe 20 years or so, this idea of what I would call scalable transient expression has become an ever increasing and important tool, and it's become becoming more and more sophisticated and advanced in terms of being able to produce recombinant proteins. And the real key idea and feature for that is that the transient expression allows us to transfect a large number of proteins all at once and do it very quickly so that you can generate proteins within a short period of time rather than taking many, many weeks to months in order to obtain your protein. It literally, from the time that you have your DNA construct in hand, it can take you a week or so to generate your protein, and then a short period of time afterwards to do the purification to get your protein in hand. So transient expression has become a very powerful tool for this. And what I'd like to do then in this presentation is talk about some of those fundamental principles and the elements of transient protein production. And here I show in the slide what I think are really, the key elements in there. Obviously, you have to have the cells, and you have a media to grow your culture of cells. You need a reagent there to deliver your gene that's expressing a protein of interest into those cells, and obviously you have the gene itself in an expression construct. So those are sort of the fundamental elements. And what I'm going to really talk about here, now there's a lot that can be said, and probably for a topic for a different presentation on expression constructs and how to optimize your gene and that construct to get the maximal expression possible. But I really want to talk to you today is about the cells, the media that you grow the cells in, the transfection reagent, etc., because those three elements really have to work together in a very coordinated fashion to get the best optimal results from your system. And so I want you to come away from this presentation just trying to think really about your cell culture and what's going on in the cell culture as you start to manipulate these cells in order to get them to produce the protein that you want. Okay, so with that, then the second part of the introduction is talk a little bit about the cell lines that typically are being used for transient protein expression work. HEK293s, they're human embryonal carcinoma cells. Those are frankly the workhorse, the main cell line that is used for transient protein production. There's a variety of reasons for that. Some of it just historical, but really it's because over time now it's been shown that the HEK293 or 293 cells are just a very good cell line for this type of work. They've been adapted, and many different varieties of 293 cells have been adapted to suspension culture, which lends to the scalability that you need in order to either transfect a small number of cells in a small volume of culture or a large number in large volumes in order to get the amount of protein that you need. They are also just are very good at taking up DNA that is transfected into them and then expressing a good amount of protein from those. So those have really been far and away the vast majority of the work has been done in 293 cells. CHO cells, on the other hand, have been of high interest, obviously, just because of what I had said at the beginning there, and that there's so much interest in recombinant proteins because of this idea of using them as biotherapeutics. Excuse me. And certainly for most biotherapeutics, when it comes down to actually producing those proteins in order to for clinical use, in order to put them into patients, many of them are being manufactured now in bioproduction settings using Chinese hamster ovary or CHO cells. So to be able to use these CHO cells as well in a transient expression application very early in the process, to be able to express your recombinant proteins in CHO cells can be of great value, because if you are looking at being able to eventually manufacture them in CHO cells, it'd be nice to know as early as you can how those proteins behave, how those proteins, what type of activity they are, the type of characteristics they may have by using the same cell line or same type of cell, CHO cells. If you do that if you do the initial work in 293 cells, most of the time that's fine, but there can be differences in the way the proteins are either the conformation of the proteins, or certainly definitely in the post-translational modification of those proteins when they're made in 293 cells versus CHOs And so that can lead to some differences in how the proteins react. So as you're screening through for biotherapeutics, it may not be exactly the same as a type of protein that you'd make further on. So those are really far and away. I would say that certainly the vast majority of the recombinant proteins being made by transient expression are being done either in 293 cells or then in a smaller percentage in CHO. However, I think that over the last few years, the work in CHO cells has been really advancing, and now more and more of it, more and more proteins are being expressed in CHO because it's become more feasible. And I'll go over that a little bit more in the next few slides. So this slide here just shows some of those cell line trade-offs that you have between the 293 cells and CHO cells. Both of them are very good at being adapted into suspension culture. That makes these transient expression systems very scalable by being able to transfect either small or large volumes of suspension cells. Two of the really key differences that I'm highlighting here in red are what really are key considerations for thinking about when you're using 293 or when you're using CHO cells. Okay, one is the growth rate. The 293 cells actually grow at a significantly slower rate than CHO cells. The typical doubling time of 293 cells in suspension culture is around 24 hours, whereas for CHO, they tend to double at around by 16 to 17 hours, so much faster. And as it's played out, it's actually become evident that the slower growth of 293 cells actually in this particular application for transient work is probably a better feature, and I'll talk to you a little bit more about why that is, that slower growing cells works better. The other one is about transfectability, and 293 cells are highly transfectable. It's fairly easy using a variety of different types of reagents or strategies to transfect 293 cells. CHO cells, on the other hand, typically are more difficult, more resistant to taking up foreign DNA. And so it's been, I think, a lot of the advances in the last few years have really been around this idea of how can we efficiently transfect CHO cells for transient expression work. And as we've been able to solve for that problem, that's really helped to increase the usability of CHO cells for transient expression and the yields that we can get out of these transient expression systems. Here I'm showing what I would call sort of the classic sort of profile of expression in transient 293 versus CHO. Particularly at the very top there, I'm showing a number of antibodies, some human and rabbit antibodies, and even the bispecific there. And you can see that for what I would call standard methods that have been used for quite a few years on 293 and CHO, that there's a substantial difference, that 293s just have been much better at expressing proteins in this transient expression application. You can see on the very top there, this is a model protein that we use quite a bit in our work. It's a very good expressor. And then again, what we call our standard 293, which is represented by, for example, for Thermo Fisher, our FreeStyle 293 systems that have been out and available in the market now for well over 15 years. You can get pretty good levels, maybe up to a couple hundred milligrams per liter, and certainly it's about four-fold lower in CHO cells, and that's pretty. That's not unusual to see that type of a difference in yields. Now, be that as it may, if you look down at the bottom three, factor IX, human growth hormone, and EPO, I listed these proteins in this particularly because even under the conditions where the standard CHO, where that transient system for being able to use CHO cells is not quite as powerful and as efficient as 293s, for certain proteins, CHO cells just have always been able to produce more. So for these three in particular, and there's quite a few other examples there are, but that the CHO cells produce more. So there's definitely a difference for just a cell line difference in what proteins might express better in one cell line versus the other. But in general, over those many years that the 293s and CHOs have been used, 293s have typically been able to express much more. So for us at Thermo Fisher, as we've worked on our transient expression system, certainly yield, even with 293s where we were seeing pretty good yields, has always been a need to get to even greater yields. So over the last about 5 years or so, we've been developing systems that has really helped to boost up the yields coming from transient expression systems. And a lot of it has to do with this idea and thinking about the cells, the media that the cells grow in, and the transfection reagent that's being used for this. In order to increase yield, one of the key strategies that has become more and more frequently used is this idea of going to what we would call high density systems, where you can put many more cells per mil of culture into your transient expression system and then being able to transfect at that higher density. And certainly since the cells are really the protein factories that are producing cells, if you can have, let's say, 2 times or 3 times more cells in your culture and then are able to efficiently transfect those cells, which in and of itself is actually a key aspect of this. But assuming that you can efficiently transfect at similar percent transfection efficiencies, those high density cells, then you should be able to get more protein out of it just because you have that many more cells in culture. Now there's many sort of considerations and aspects of it. If you have high density cultures, that means your entire media system and that culture system has to really be able to provide the nutrients and other key factors to sustain the viability and expressibility of those cells, even when they're becoming much more crowded and have many more of them in there. It puts greater demands on the culture system itself to have these high density cultures. And similarly for the transfection reagent, it puts greater demands on being able to transfect many more cells per mil of culture in there. You can imagine that if we were able to increase the density of your culture, but your transfection efficiency fell off, that for example, if you had, let's say, a 3-fold increase in the number of cells, but your transfection efficiency dropped by 50%, then you're really not gaining as much of the benefit of having 3 times more cells in culture as you could if you were able to also increase and improve the transfection to maintain the high efficiencies. So these 3 areas are really very well tied in together and are very important in this idea of transient expression work. Let me show you that now. So for Thermo Fisher, we have developed our high density 293 and CHO systems. They've come out over the last few years. Our Expi293 system, our high density system for that we developed and came out with about actually almost 5 years ago at this point. And our high density CHO or ExpiCHO system, we just recently launched in September of 2015, so about a year and a half ago. And here I'm just showing you now the results that we've taken the same proteins that I showed in that panel a little while ago, and showing the results of how much more expression that we can get out of these. I'll just show very quickly here. You can see now with our Expi293 high density system, that same human IgG that's been sort of our high water mark and our reference for protein expression, we can express that at on a very consistent basis at about 1 gram per liter of protein. That same protein in our high density CHO or ExpiCHO system, oops, I'm not very good at drawing here on the board here. We can, that same one now, using the same exact constructs that we use for our 293 systems, when we transfect that into our ExpiCHO, we can generate up to 3 grams of that protein per liter. So it's a huge advance in terms of transient expression to be able to get to these high density systems, and then coupling, again, a media that really helps to support the density, a transfection reagent that helps to maintain the high efficiency transfection under those conditions. You can see a significant improvement in yields across the board, obviously for that model protein as well as for many other proteins, a significant jump in expression. So here are, I'm not going to really go through this, and probably later on this presentation will be archived and you can take a look at some of these numbers. Just a few parameters about these systems, the densities at which we work at there, the maximum working densities, etc. Probably here the main thing to point out is that the second line here, density at transfection, our Expi293 or 293 systems, we're now transfecting at about 3 times more cells per mil than our original FreeStyle 293 system, and for our ExpiCHO, we are able to actually transfect that at 6 million cells per mil, so 6 times higher number of proteins. Let me start really getting into now the things that I think are important to consider and think about in terms of how whatever system you're using, whether it's our sort of the more standard classic systems or these higher density systems, and whether it's from us or from systems that you have implemented yourself from a variety of other sources. I think the underlying principles that we're going to talk to you about next are very important. So really, if nothing else, what I want you to take away from this presentation is this idea of cell health and the culture and what's really going on there. Because frankly, that is the key to really getting the optimal, maximal performance you can from whatever transient expression system you're using. Think about the cells. You want those cells to be as healthy and robust as possible. There's a variety of factors that play a significant role in that. So most of them, or I think many of them, I point out in this slide. Certainly the particular type of cell, as I mentioned already about the rate of growth of the cells, that needs to be taken into account. The media, the formulation, whether or not you have fresh or not as fresh media in there, the age of your cultures, the amount of stress that the cells are undergoing as they're being cultured and in this sort of suspension culture system, oxygenation, etc. These are all key parts of it that you should at least have in your mind and think about as you're using these cells for transient expression work. And then this idea of a transient culture expressing protein. So when you do a large-scale transient run, you're transfecting a number of cells, either starting at the classic cells, which are about 1 million cells per mil, all the way up to now 6 million cells per mil with our ExpiCHO system. Okay, now when you do the transfections, not every single cell is going to be transfected, and certainly they don't all express at the same level. Very different from a stable expression system there, where the cells are all integrated with the gene. So you'll have this mixed population of what I would call productive and non-productive cells in your culture, and that's very important to think about because there are two different you essentially have these different sort of characteristics and populations of cells. The non-productive cells are not only, let's say, not contributing to the yield, but they actually have a active sort of negative contribution to the culture, because typically they grow faster in your culture and they will metabolize the nutrients in your media quicker. So the more non-productive, non-transfected cells you have in culture, the quicker the media is exhausted, the shorter time that the productive cells will have in order to express their protein. And over the next few slides, I'll just show you sort of in a little bit of an exaggerated format, sort of the dynamics of what goes on in those cultures. So you start off with this, you can see that the picture of, the number of cells you have being transfected. And for most of these systems now, for example, our Expi293 and ExpiCHO, we can achieve transfection efficiencies of around 80 to 90% sometimes better than that, but I'll say that those certainly are very consistent and reasonable numbers that you should be seeing, at least in terms of transfection efficiency. So over the next few days, what's going to go on is you have these differential growth rates between the non-transfected or non-productive cells and the productive cells, and you'll start to see that in your cultures, more of the cells will will not be contributing to the recombinant protein that you have, if you just allow them to continue to grow at these differential rates, so that eventually over time what you'll see is you're going to crowd out the productive cells with the non-productive cells. The nutrients are going to be exhausted, and the overall the expression may not last as long as you'd like, and so you don't get as much protein out at the very end. So this sort of dynamic I've exaggerated in these photos, but it's something definitely that you should think about and consider whenever you're doing a transient expression run and when you're setting up your systems. To think, how can you manage this and make sure that your cells are doing well. So the first thing we always do when we do that is really think about, okay, what are the things that I should be monitoring and thinking about? So for us, it's very routine, and really we want to be very regimented about this. We want to make sure that we're monitoring the cells. We're checking the growth rates, to checking the number of cells in culture all the time, to be able to really understand what's and see what's going on and see that in culture. Now I'll talk a little bit more about, we need to maintain the cells in what we call early to mid-log phase growth. That's when the cells are most productive for protein expression. And so knowing, for example, at what point your cells will reach maximal density and start to move into static phase is important, because then that gives you an idea of what the working range and where your early to mid-log phase growth will be for your culture. And then you establish a very routine passaging culture. For us, we typically do it 2 times a week. Rather than 3, just because that introduces less of that lag phase whenever you're doing a media exchange, or actually in our case typically just dilution with fresh media. But the important thing here is just to be very routine about doing it on a consistent basis, but monitoring the density of your cultures to make sure that they're within that early to mid-log phase growth. And we also monitor the viability, the percent viability of those cultures, and the number of passages we go, and I'll talk a little bit more about that in a second. Also, to avoid cell clumping, there's a variety of different ways that you can avoid cell clumping. Sometimes it's adjusting your agitation rate for your cultures. Other times it's making sure that you just do some work as you're passaging the cells, to perhaps titrate them up and down a little bit in your pipette to try to break up any small clumps. Or sometimes you might, if you're starting to see some significant visible clumps, to let the culture sit for a second, for a little while, let the clumps fall to the bottom, and then sort of carefully take off the cells off the top of your culture. A little bit of clumping is not probably a huge concern, but certainly once you start to see a lot of clumping, typically you're going to have to go through these, and a lot of times for us we just get rid of that culture and have a fresh culture started to avoid clumps. Okay. Here I'm showing again for monitoring of your cultures. It's very important that you do the counts and understand what the doubling time of your cells are. It should be pretty consistent from passage to passage. If it's not, then there's something that you might need to look at and address in your culture system, how the cells are being handled, or the equipment that you're using, etc. And here I'm just showing sort of typical. These are for FreeStyle cells and FreeStyle media. But the numbers hold fairly true as well for our newer high density Expi systems. 293 cells, you can see here, for over 10 passages, the calculated doubling time of those cells averages out here to be about a little bit more, about 25, 26 hours for doubling. For Expi, our high density Expi293 systems, that drops a bit down to about 24 hours. For CHO, it's been the same, whether it's for our older FreeStyle or our new Expi ExpiCHO system, that they double about every 17 hours. And you can see that they're relatively consistent from passage to passage. Now, there have been times when we've all of a sudden noticed that a culture has started to increase in the time it takes for that culture to double. And when we see that happening, then we start to think that there may be something going on in there, and much of the time we'll just take those cells out of our workflow and put in fresh cells for that. And in addition to this doubling time, we're monitoring, as I said, the cell viability. And typically, most of the cultures with either 293 or CHO, you should be able to get at least 95% viable cells consistently all the time. For our Expi systems, even for our FreeStyle, typically we're seeing that more in like the 97, 98% viable cells. And for us, that's it's good to see that and that it's consistent. If you get these two parameters, and you're seeing that very consistently, the doubling times, the viability of the cells, then the likelihood is that you're going to get much more consistent results in terms of the protein yield from them from one time to another. Okay. Here I'm showing, I'm not going to spend any time on this, but again, you can come back to this when this presentation is archived to take a look at this. For anybody who doesn't already have or know this, this is the formula in order to calculate the growth rate of your cells, and so this is what we use in order to determine those. Okay, so now by being able to maintain a very consistent, steady culture of the cells, having the consistency in that doubling time and the viability, etc., it's very important. Another aspect I talked about is about the age of your cultures. Here we did an experiment. This was with our FreeStyle CHO system a number of years ago. What we did here was we took FreeStyle CHO and thawed them out at different various times. So by the time we did this particular study, our oldest cultures had been thawed out about 12 weeks previous. So those are the bars in blue, and our newest cultures had been just thawed out 2 weeks prior to this. So those are the bars in black. And you can see what happens. This is post-transfection, the expression of IgG, and at 1, 5, and 6 days post-transfection, the yield of that IgG that we got from the transient expression run. And you can see that for the older cultures, the ones certainly at 12 weeks, there's a substantial reduction in the amount of protein that we're able to get. So certainly over time, for a variety of reasons, the cells just lose more of their production capabilities for that. So it's something to monitor. Typically for us, we go out about 2 to 3 months of culture of using the same culture for our expression runs, and then by the time they're reaching close to that 2 or 3 months period of time, then we'll already break out fresh cultures and just switch over to them to really maintain the maximal yields that we can from the cells. You could probably go out a little bit longer with 293 cells than with CHO because of the fact that they grow slower, and so over that period of time there's actually fewer doublings of those cells than with CHO. So that typically will allow you to use those for a longer period of time before you have to switch to a new culture. The other thing is, as I mentioned before, about the being in early to mid-log phase growth and not allowing the cells to really get to a lag phase or a static phase where they've grown to their maximal densities and are just sitting there. If you do that, then it really does impact the yield, even if you then dilute the cells back down and get them back into what I call what we have as early to mid-log phase growth. For this particular experiment, what we did was we took the cells and did passaging, but for the ones in the blue line, we only diluted those cells down to about 1 million each time as we passaged, and then allowed them to grow for another 3 to 4 days. And so they would at that point have hit the static phase, and then we passaged them back down. And whereas the other ones we passaged down to lower densities, and so they maintained a little bit more of their early to mid-log phase growth, and you can see the impact of that the cells that were getting up into that lag phase just do not produce as much protein as those that are maintained in the log phase growth. Here, what I'm showing here is this is where, again, having high density cultures in a media that can really support much higher growth rates or density, sorry, of the culture can play a role. I'm showing here Expi293 cells growing in Expi293 media compared to 293 cells grown in a variety of other medias. You can see that the maximum density that the Expi293 cells get to is much, much higher. So that means that early to mid-log phase growth is in a different working range, and hopefully the animation, so yep, here shows what happens. So the working range in that up to mid-log phase, you can see are shown in those brackets here. And for Expi293, that allows you to really maintain those cells up to around 5, maybe 6 million cells per mil, and then passage. So typically we passage down to maybe about 300,000 and then go up to between 4 and 6 million cells per mil. Whereas with these other, our older systems and other systems, that working range is significantly reduced, and that we can really only grow those cells up to maybe about 2 million cells per mil. And then we do the transfections at 1 million cells per mil, which is in the middle of that range, versus doing a transfection at 3 million for our Expi293. Okay, in order to be in that right in the correct working range for those cultures. Here for our CHO, our ExpiCHO system, those cells now with the highly enriched media formulation and the adaptation of those cells to the high density cultures, those cells will grow up to over 20 million cells per mil at maximum. So that means that the working range, that early to mid-log phase growth, is significantly extended, and we can grow those, and typically you're going to passage those cells up at, by the time they reach about 6 to 8 million cells per mil, and then drop them back down and maintain that range. And that allows us then to do the transfections at higher density here at about 6 million cells per mil. Again, leading to higher yields, protein yields that can come out of them because of the higher density. There's actually other benefits for the media. The long-term adaptation into this highly enriched media also makes these cells, both the ExpiCHO and Expi293 cells, provide higher per cell productivity. So the picograms per cell per day for these cells in this enriched media under these conditions typically is higher than the picograms per cell per day that you can achieve with what I'd call standard density systems. The protocols for being able to use, when we have these high density cultures, then it does make it a little bit more challenging to do the transfections and maintain the viability and growth of the cells, or not growth, but the viability of the cells over the time post-transfection. I'm showing here, I won't go through the entire thing, but sort of our standard protocol for transfection and handling of the cells for our ExpiCHO system. And the thing to consider here is we have not only a transfection reagent to be able to transfect as many cells as possible for ExpiCHO, typically around 80% transfection efficiency, but then there's other aspects of the system in order to manage that what's going on with the cells. So typically for us now, the strategy that we've started to employ is to think about using how to basically shift the metabolism of those cells, because they're growing. We are using those, expanding them in culture, so they're in sort of a growth phase. We keep them in that early to mid-log phase growth for that. But then once they're transfected, ideally what we would like to do is slow those cells down, or actually even potentially get them to stop growing because of the dynamics I showed you before with the mixed population of cells, and shift them over so that instead of spending their time and energy on doubling, they're spending most of their time and energy on expressing protein. So the transfection reagents that we've designed, we have what we call an enhancer, transfection enhancer, and then also a feed. And all of those things are designed to both to maintain the health of the cells and also to shift their metabolism so that they're no longer really growing very much and using most of their energy for expressing protein instead. And we also, for our CHO system, because CHO cells actually do have an ability and are sensitive to temperature. So by dropping the temperature of the cells in culture as well, you can slow them down and shift their metabolism from growth over into expression mode. So all of those things play a role now in these more advanced transient expression systems, to think of how we can take the cell lines post-transfection and move them over into a full expression mode rather than in growth mode. Another thing to consider is, and is important, is the kinetics of being able to make your DNA-Transfection complexes there. Now, it's really important to understand that when you take your DNA and put it together with any of the cationic type reagents that are used for transfecting, whether it's the cationic lipids that we produce or PEI or others, there's typically what happens is, as you take the DNA and add it to your cationic reagent, you form an electrostatic complex, and over time that complex actually will continue to grow in size. And so once it gets very large, the transfection efficiency of those complexes goes down. So timing is important. Here I show you for actually, this again is our FreeStyle CHO system, but it applies to pretty much any system there. What happens when you allow the complex to sit for 10, 20, 30, 60 minutes? And over time, in each of the particular cationic reagents that you can use for transfection are going to have a slightly different temporal pattern for this, but over time you can see that the efficiency of that complex is reduced when you do this. For our latest ExpiCHO system with the Lipofectamine transfection agent, you can see that we actually don't want to allow those complexes to sit for more than 5 minutes together, the DNA and the transfection agent, before you apply them to the cells. Once you go out to 15 or 30 minutes, you'll see a significant decrease in their performance when you put them onto the cells. So that's definitely something important to consider, no matter what type of transfection reagent you're using in the system. And then lastly, if I really try to bundle together some of these concepts, what you should be able to see once you transfect the cells is a very reproducible sort of behavior and pattern for what's going on with the cells. And again, we monitor this by looking at the viability, percent viability of your population, as well as the viable cell density over time. So here I'm showing with Expi293 culture, the culture kinetics post-transfection. So we transfect that here day 0 and then monitor over the next 7 days what's going on. You can see how the viability starts off very high at well over 95%, and then for the first several days we'll maintain pretty good viabilities up above 90%, but then start to drop and accelerate in decreasing in viability. So that typically by the time you harvest, which is somewhere between 5 and 7 days for most folks, you'll see viabilities around 50%, or actually even going a bit lower down into 40%. If you see it much lower than that, if you're harvesting at, let's say, day 6 or even or so, and you're seeing below 20% viability, then that's suggesting that there was probably something in your culture or the way that these cells are being handled that made them a little bit less robust. And typically, you'll see a reduction in yield if you see that. So knowing this viability and viable cell density curve for your particular expression system under your standard conditions in your incubator is very important, because that really helps to give you a gauge and monitor what's going on. Here you can see for viable cell density for Expi293, they start off at around 3 million cells per mil at transfection. Then they will expand for the first several days till they get to somewhere between, let's say, 6 and 8 million cells per mil. So they slow down, and certainly if it were non-transfected cells, they get to that density much quicker. But they do slow down, but they will continue to grow, and then they'll sort of peak and then start to sort of decrease again as the viability continues to drop. So these are very common, what I would call prototypic viability and viable cell density profiles and curves for Expi293 system and 293 systems in general, although the densities, it'll be different ranges of densities that you'll see depending on the particular system. So these are things that you want to monitor. If you see that they're following what I would call the prototypic pattern, and they're either not growing too high in density or they're not dropping in viability too rapidly, then probably your cultures are going well and you're getting maximal performance out of your cells. And you can see this, it should be, you can monitor these and it should be very scalable. Here I'm showing at the top there Expi293, and at the bottom FreeStyle 293. The red curves are wave runs being done at around 8 liters of culture, and the blue curves are small shaker flasks. I think in this particular case about 40 mils of culture in there. And the viable cell density should look very similar no matter what scale you're at. So this is an important concept for scalability of your systems, that as you scale, you're probably going to need to adjust some of the parameters, either the agitation rate, etc. And by monitoring these things, that will give you a guide to how you need to sort of adjust those culture conditions in order to basically try to replicate the same viability and viable cell density curves. If you can do that, then typically your volumetric yield, in other words, the milligrams of protein per liter of culture, should remain fairly consistent across different culture volumes that you're using. All right. Here I'm showing the kinetics for ExpiCHO, which are significantly different from 293 cells for a variety of reasons. For CHO cells, and probably also because of it's a more recent system that we've developed. We've been able to apply a little bit more advanced techniques in terms of our culture conditions, or the media, the feed that we add to those cells. We can maintain viability of those cells much longer. So you can see on the left-hand side the viable cell density and the viability curves. I'll start with the viability curve on the bottom left there. Start at, again, in the high 90% viability, and even out for almost 2 weeks now, we can maintain the viability of those cells at over 80%. Okay, and part of that is because we really have been able to figure out methods because of the transfection reagent, the enhancer, the feed, and also potentially, depending on which protocol you use, the drop in temperature. We can maintain the viability, the viable cell density, and shift those cells from growth rate over to basically being in static. So at that upper left, you can see that the cells start at about 6 million cells per mil. They'll grow up to potentially around 10, maybe a bit more than 10 million cells per mil, and they basically don't grow anymore beyond maybe the first couple of days, and they'll just maintain a fairly consistent cell density and high viability. And that allows these cells then to just continue to pump out protein over a substantial portion of time, and this is what makes for the high yields that you're able to achieve now in our ExpiCHO systems. Typically, what we've seen in various scenarios where somebody may not be getting quite as good yield is we'll see basically that as you get out into the later phases, that the viability has substantially dropped. So that's the curve on the right-hand side compared to the graph on the left-hand side. If you see that where they drop substantially below 80%, that's suggesting that probably, again, it's adjusting the culture conditions. So whether it's RPMs or rocker speed, an angle, or whatever is required to agitate your cultures. There may be either too much agitation going on, creating too much stress for the cells so that they drop in viability, or potentially also maybe not enough agitation to get enough aeration into those cultures. So it's by taking a look at these things, seeing that, then you can start to make assessments of what you need to do to optimize your culture conditions in order to get the maximal yields. These are essentially very important, and really the emphasis today is monitoring your cultures, understanding what the dynamics of that population is looking like over time to be able to achieve the highest level results. So that really is the key takeaway I hope that you get from this presentation. So just as a summary, 293 cells, CHO cells are really the most common cells used for transient expression. The growth characteristics of 293 cells and CHO cells are very different, so you have to take that into consideration about the difference in growth rates, and also the idea that, for example, 293 cells, we would consider them a bit more of a system where the cells will actually are pretty robust and just continuing to grow, and then will sort of hit a maximum density and start to die off. So it's continuing changing over time. Whereas for CHO cells, we have systems that are able to allow the CHO cells to really shift in their metabolism, so that they go from a growth phase really into more of just a maintain the same density but maintain high viability over time to be able to produce more protein over a substantial portion of time there. Over the last several years, because of the introduction, for example, of the ExpiCHO system, really now I guess which cell line produces more protein has really changed right now for us, at least. We see, and not only internally but with many of the users of ExpiCHO outside of ourselves, we're seeing probably on average about a twofold increase in yield on ExpiCHO compared to the best 293 systems that are available out there. The culture conditions, which hopefully I've spent enough time on, are key critical parameters for what you're doing. So really being able to monitor your growth, the viability, also really understanding and monitoring your seed cultures and making sure that they're being very consistent in how they grow and how they passage, and checking on the number of passages is very important for this. For transfection, the transfection technologies, whether it's ours or others, typically are pretty robust. The main things is paying close attention to timing of that transfection complex formation and not allowing those to incubate for too long before you add it to the cells in culture. Otherwise, you can get some decreases in the transfection efficiency. So with that, I'm pretty much done with my presentation for today. Thank you very much for your time today and listening to this. And I'm happy to, we've actually got plenty of time, although we obviously don't need to take it off, to take any questions that you might have. And if not here, then you can certainly send in questions later on, and we can answer them offline as well. So with that then, Kristen, I think I'll turn it back over to you.
Thank you so much, Dr. Chiou, for that informative information. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of the questions as we have time for. So let's get started. Our first question is, what is the difference between Expi293 cells and regular HEC293 cells?
Kristen, for that question. Oh, thank you, Kristen, for that question. Yeah, so Expi293 cells and are essentially what I'd still call wild type HEC293 cells. We didn't do anything in terms of engineering of the cells. What we really basically did with them is we took our FreeStyle 293F cells and we did long-term adaptation into this highly enriched media that we developed, the Expi293 media. And it basically was more of just an observation phenomenon that we observed, with those as we continued to passage the 293 cells in the Expi293 media. They started to change somewhat in their characteristics. They started to grow a little bit faster, as I showed you in one of the slides, as well as they started to produce more protein. So the picograms per cell per day for those actually ended up increasing by about 30 to 40% compared to I guess the early adaptation of those cells into the XP media. And then after that, it sort of stabilized and leveled out. So then we banked them at that point. So there are really no differences genetically between the Expi293 and what I call standard HEC293 cells. There probably are differences from a point of view of really what gene transcripts. We've done some work to know that some of the genes have increased and other genes have dropped in terms of their expression levels, but in terms of the genotype, it's the same. Okay, next question.
Thank you, Dr. Chiou. Okay, for our next question we have, apart from using low passage cells, maintaining a high cell viability, splitting the cells according to the expected cell density ranges, maintaining appropriate shaking conditions, or 125 to 130 RPM, and avoiding temperature shifts, do you have any advice in order to avoid XP-CHO cell clumping?
Can you hear me? Okay, sorry, I might have started before. In terms of clumping, the nice thing is that we've actually found that with the XP medias and that long-term adaptation of the cells, they don't tend to clump as much, but I can certainly imagine that in some instances you can get clumping. So again, it's really a measure of making sure that your agitation rates are in the right range. There's always a little bit of a balance because without enough agitation you can get more clumps, and certainly the cells may not be as well suspended in culture. But with too much, then you're creating too much stress on those cells. So finding that sort of that ideal range, depending on your particular shaker or whatever other type of culture vessel you're using, it can vary from one person to the other depending on their equipment. Now, the other thing that actually will have an impact on clumping, which we don't talk about as much, is temperature. And when if particularly, for example, if you have shake flasks that are sitting on a shaker, that shaker does give off some heat. And depending on how old or new that shaker is and various models, you may have more or less heat exchange going on there. So we always actually, when we install a shaker in there, we'll put in a flask with some liquid water, whatever, and a thermometer, really just to check what the temperature is right at the surface of the shaker. CHO cells in particular, although I think 293s can suffer from this to a certain extent, as the temperature goes up, they can clump more. For CHO cells, we actually like to, 37 degrees right there at that in the liquid, right sitting on the shaker platform, is the maximum we want to go to. In fact, a little bit below 37 is probably fine for the CHOs. They can withstand that. 293s probably have a little bit more flexibility, but you really don't want to go much above 37 degrees for those, and that can cause some clumping.
Okay, thank you. Our next question we have, what is the effect of antibiotic or antifungal agents on protein expression?
Okay, so typically, for these cells, we try to understand and monitor all the different stresses that are impacting those cells in the culture. So we've done some tests, particularly antibiotics. I don't think we've really tested antifungals too much. You know, for us normally, we avoid using them at all, and typically, you know, we're able to maintain our aseptic conditions so that we don't have to use them. Now, it is possible, we have tested, and we do know that we can use a bit of antibiotics in these cultures, and normally they're fine. You have to consider all the different stresses that are going on, and particularly ExpiCHO cells are more stress sensitive. So if you've got the right RPMs, agitation rate, and other things, you're passaging them well, the cells are very robust, then taking a little bit of stress from antibiotics or potentially an antifungal agent, probably okay. But if those cells are either having either too much agitation or maybe too little oxygenation, or are already under some stress, then adding that additional stress may have an impact. So part of it is making sure that your culture conditions really are just as optimal as you can, and then if you do that, then adding the antibiotics, etc., shouldn't be too much of a problem.
Okay, we're almost out of time, Dr. Chiou. This will have to be our last on-air question. Would a temperature shift be effective for HEC293 in increasing protein EXP?
All right, thank you for that question, and actually that's a good question to end with little time, because it's actually a pretty quick answer. We've tested that. We've had a number of other groups try to use that as well, and for basically everybody's come to the same conclusion that at this point 293 cells just don't seem to react the same, and the temperature shift doesn't really do much. It doesn't really help them. They just chug along at the lower temperature just the same as at high temperature, and you probably just it either has no impact or actually it can just decrease the performance of the system. All right, thank you very much again. Thank you everyone. Thank you everyone.
Thank you so much. I would like to once again thank Dr. Chiou for his presentation, and I would also like to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.